but don't forget to do that, please. It's due Friday. And then I'll talk a little bit about that one and the other one Friday in class. Because the following Friday, not this Friday, but next Friday, we have an exam. All right, so it's gonna start with the photosynthesis stuff, because we covered through plant growth and propagation. So now we're into the material and photosynthesis. And then roots, stems, and probably leaves as well. Okay. So make sure you're keeping up on that. So stems, we got to think about stems. So we looked at stems the other day. And so if a botanist talks about wood, what they're really talking about is secondary xylem. And that's what's being formed if you have a dicot tree, for example, that's growing over the years. And you can definitely count the growth rings if you want. And we talked about last time how when the plant is going through its seasonal growth, they will grow a little differently in the sense of what types of xylem they will start to form. So under really nice watery conditions where the soil is nice and moist and there's a good amount of soil or water they can call upon in the soil, then they'll use those vessels. They'll lay those down as they're growing to get access to all that really lovely water very efficiently. And then in the summertime, the summerwood, they will put down the narrower, smaller in diameter tracheids to help prevent any kind of their version of an embolism, which I'll talk about in a little while here. So often, when I was a grad student up at Michigan Tech University, we all lived in this big mansion in Hancock. And most of the other people that were renting there were forestry students. And so they would talk about spring wood versus summer wood. And so some of these terms that I'm showing you are more how foresters would talk about some of the ideas, but it's still relevant to what, so if you've ever wondered what some of these terms mean, like they'll talk about heartwood, and then they'll talk about sapwood. And so the heartwood and I'll show you a picture of this. It's when you look at a tree trunk in cross section, you can see a darker patch more toward the center where there's been a lot of tannins that have been produced and secreted in that area, or resins and gums. And so that area of the xylem pretty much isn't functioning as water transport. They're just being used for support. And the tannins and so on are really good at protecting that portion of the tree against attack by fungi or insects. And then the so-called sapwood is the active area of xylem is actually being used for mineral and water uptake. So going back to some images, something like this. So here, you can see that looks like 3 years of growth approximately, and you can see the vascular cambium area that's delineating the wood, the secondary xylem, from the phloem to the outside. And this is a fairly young sapling. This one is a better image showing the summer versus spring wood. And so you can definitely see the larger vessels. And then the smaller summer wood. And then the next year in the spring, they put down vessels. And then over the summer, they put down tracheids, vessels the next year, next spring, and tracheids. So this growth ring, you can see is much wider. So that must have been a good year for that sapling. It must have had lots of adequate water and minerals and enough sunlight. And so it has a nice growth period there. Whereas then you look at the next year, not so good. So you can tell a lot from looking at tree rings in terms of how wide they are, how many vessels got laid down versus tracheids. That can really tell you a fair bit. And so obviously, if you take a full cross section of a tree trunk, you've killed the tree. But you can safely take core samples into the tree trunk and study the core samples without killing the tree. Okay, so now after looking at this picture, now let's look at another picture. So here, do you see obvious big differences in summerwood versus springwood? Not obvious, right? Not like that. So if you see something like this, then you should start suspecting, I wonder if I'm looking at a gymnosperm, you know, like if no one told you what it was and just said, hey, look at this. So here, remember now, pine trees, gymnosperms, they're more primitive than the angiosperms. They don't have vessels, except for some weird exceptions, which I'll talk about. So by and large, they don't have these nice vessels and then some tracheids, vessels, tracheids that they're laying down. It's pretty much tracheids. They can vary the size a little bit, but not dramatically. So if you see a more homogeneous visual like this, when you're looking at tree rings, chances are it's a gymnosperm when you are looking closely. This is just if you take more of a longitudinal view of it and look at it, and they're just, you know, pointing out the annual rings here. So here you can see the heartwood versus the sapwood. So here you can see this one, we have a really big area that's been impregnated with secretions, and it's often, you know, if you've looked at the structure of tannins and things like that, they're quite nice. Fungi have a hard time getting access to this area of the tree because of all those secondary metabolites that are getting laid down. And then the lighter area is the so-called sapwood, where you have active water and mineral uptake. And then sometimes you hear the term hardwood versus softwoods. And if you've ever wondered, then we're talking generally when you say it's a hardwood, then you say it's a dicot tree. Softwoods tend to be gymnosperms. Okay, so again, those are forestry terms that you see or hear people talking. All right, now if the tree makes it past seedling and sapling stage and it starts to form bark, we need to talk a little bit about that. So bark is really nice for protecting the stem tissue. And the formal definition of bark is that second bullet point. All tissues outside the vascular cambium equals bark. All tissues outside the vascular cambium equals bark. So that includes the secondary phloem. So part of the bark is actually phloem tissue, which I always thought that was really weird, but that's how they define it. And then what we're going to see is as the bark is starting to form, the epidermis is going to go away, and you're going to see a periderm taking the place. And if you look at the periderm, there's different parts to it. So there's a cork cambium, which is meristematic tissue. The old term for it is phellogen. I don't like to use that, but you're going to see some terms probably in lab on the slides where they might say that. So you do at least need to be familiar. But the cork cambium, we have a vascular cambium in the stem that gives rise to secondary xylem and secondary phloem. Now we have a cork cambium further out toward the perimeter, which is another meristematic region. And so it's going to produce the so-called periderm region and cells. So what's going to happen is similar to the vascular cambium. The cork cambium is going to go through cell division, and it's going to produce one type of cell to the inside and another type of cell to the outside. And so to the inside, well, I guess I have it to the outside first. So to the outside, they produce cork cells, which at maturity, they die. So they have secondary cell walls. And the cell walls are very heavily impregnated with suberin wax. So suberin is a type of wax that you tend to see in bark, and then also around roots. Sometimes they'll be suberized. And then they call them secondary cortex cells, which are just parenchyma cells. So I tend to think about it that way, where the cork cambium produces parenchyma cells to the inside, they're living, and then dead cork cells to the outside. You know, initially they're living, but then they'll die off once the secondary cell wall gets formed. So sometimes the older, like those slides that you'll see in lab, they might be labeled phelloderm. It's just parenchyma cells. Okay. So the secondary phloem plus the periderm, essentially taken all together, that's your bark. Bark can be very diverse visually. You can see, I'm sure just living your life to date, you've noticed that bark can look very differently. And sometimes it can be very thick, sometimes it's pretty thin. As you know, the birch likes to peel. I'm guessing many of us in this room couldn't help ourselves and wanted to peel further when you see it flaking like that. I just implore you not to unfurl it all the way around the trunk. So if you do that, if you take this and peel it all the way around and you took a thick enough section, you've committed that tree to die. And I don't think we want to do that, right? We're not the type to do that if you're taking this class. So please don't do that. I used to, when I would, go out hiking with my boys when they were young, they'd want to do that. They'd want to peel. I'm like, no. So basically what's doing, if you take too thick of a section and peel it all the way around, you've taken the phloem in that area. And so then what happens is, up here, the leaves are doing photosynthesis, and then they send the sugars down, they reach this area and there's no more phloem. So you've broken the connection. And now you can actually see the tree trunks will swell here just a little bit because all the fluid is gathering here and it can't make it because you've disrupted the phloem. So then the roots start to starve of sugar. So it's a very slow death, slow and agonizing for these trees. So please don't do that. Tell other people not to do that. And then obviously it takes a while if it's a tree, but it will die eventually. So some bark, you can see here, all these lines, these are lenticels. I'll talk a little bit more about that. Sometimes you can't see the lenticels, they're there, but you need a hand lens and maybe even a microscope to tell the difference of what's going on there. And some bark is amazing. Like that's not photoshopped. That's real. Isn't that something? In person, you've seen it?
Yeah.
Oh, good for you.
They are like that though.
Yeah, I know. I've not seen one in person yet, but I'm thinking, boy, I wish we could grow this in the greenhouse. Wouldn't that be fun? Walk in there on a snowy day like this and see that in the greenhouse. But they're tropical, so I don't know how well they would do. So this is not Photoshop. These are the actual colors. And apparently, from my understanding, they'll like peel off over time. So you can get like one tree trunk over time will give you this varying look and different colors over time. So yeah, that's just beautiful. Anyone else been able to witness this in person? So I've seen them, my friend went to Hawaii too, and she's seen them. And then I think they're also in Australia area. They're pretty common there, but I have not had a chance to see them. One of these days. So, and then there's, more boring bark like this, but again, it can be very thick or thin. Lenticels can be hard to find, but bark serves its purpose, whether it's that beautiful color or not, in protecting the stem tissue. It provides some hydrophobic, because of all that wax, and so it's nicely hydrophobic. Here you're seeing kind of a up top is like the younger stage, and then the bottom picture is a little bit more developed, and so they're trying to show how you get these layers of cork, to the outside of the cork cambium. So the cork cambium is the dark brown, and then when the cork cambium goes through cell division, it'll produce the parenchyma cells to the inside and the cork cells to the outside. And you can see how it still has an epidermis, but that will slough off, break open and die eventually, and then it'll get replaced by the true bark. So here you're seeing layers upon layers of cork. So these more squished cells are the cork. And then the cork cambium is roughly in here. And then to the, I guess it's here. Let's see, close. And then the roundish cells, those are the parenchyma cells. And so here, this one's labeled. So you have the cork cells to the outside of the cork cambium. And then parenchyma cells to the inside. And then this one is nice because it shows both. So here you have a stem and cross section. And then they've blown up a pie section of it, so to speak. So you can see the vascular cambium that's giving rise to the secondary xylem and secondary phloem. And then the cork cambium is way out here. That's giving rise to the cork cells to the outside and the parenchyma cells to the inside. So they have it nicely labeled, everything outside of the vascular cambium, that's your bark tissue. So if they're so well suberized with the cork cells being so waxy, they still need to breathe because you have phloem and parenchyma cells, those are living, they need oxygen. And so those bark lenticels play that role. So like this one, you can clearly see lenticels look bumpy and scabby, but really it's just a region, discrete region of loosely packed cells right there to help the cells get access to oxygen primarily. So it's for better gas flow. So that's the function. And sometimes you can see them very obviously, and sometimes you can't. But the cork cambium will lay down discrete regions where it's a little bit loosely packed like that. And then some fruits, apples and pears, you've probably noticed little like striations and areas on the so-called skin. And those are lenticels too, even though that's not stem tissue when you go to eat an apple, but still they have lenticels there. So lenticels are mostly on stems, but sometimes you can find them elsewhere.
Having, I'm sorry.
Oh, no, that's fine.
Too big, like lenticels on a fruit that like over oxidized.
No,
Does that ever happen?
not usually, but having said that, you know, it's loosely packed region. So fungi in particular, bacteria, that could be a way to enter. And,
Okay.
you know, here's this nice juicy sweet flesh where a lot of fungi would be like, yeah, you know, so makes sense that they have pretty small lenticels. But, you know, even on stem tissue, fungi and bacteria can get in there and start causing problems. So it's this, you know, compromise that the plant has to make. I have to breathe, but I don't want to be too big in my lenticels because then I could get attacked. So it's a game of trying to get that balance. Ooh, that's kind of blurry. But here you can see some lenticels too, some of the trees that we have around here. And then under the microscope, you would see something like this where you can definitely see it's much more loosely packed. You can see how they would have a little bit better access to oxygen. Here's another example. Okay. Then some plants, they will need to modify their stems in order to thrive in the areas that they're living. So here's some examples. So desert plants are amazing in their ability to adapt. We've already talked about cam plants and how they can do a very special form of photosynthesis. The left image are flattened stems. Some people would say, no, those are leaves, but those are flattened stems. And they're called cladophylls. That's the fancy name. I would like you to know the fancy name. And then the right, the giant saguaro cactus, that's a succulent stem where it's nicely full of a lot of water. So of course, a lot of animals in the desert want that. So they have to defend themselves really well to try not to get eaten or get tapped into for water. Here's a whole series of different things that stem tissue can adapt. So here, I'm going to go through some of these in the next slides here. So bulbs, we'll look at some bulbs in lab. And so when you look at, do I have a comparison? So bulbs, like an onion bulb, what you tend to see if you cut into one, a really modified short stem. And then all of what you're eating pretty much is leaf tissue when you eat an onion, and then the roots. You can usually see when you buy an onion. By the way, do you know the trick not to cry when you're chopping onions? Yeah, very good. Yep. I was making a chili the other morning and my son came out of his bedroom. He's like, why are you wearing safety goggles in the kitchen?
But isn't it one part, like, I think it's by the roots that produces that smell, so
Yeah.
if you save that part for last.
Yep.
Then you don't cry while you're cutting your.
Exactly. So do both. Wear goggles. I wear safety goggles that fit really well. And then the other thing too is refrigerate it, get it cold. And then leave that root area for last. And because as soon as you cut in down here, it really pumps out the volatiles. So if you do all that, the last few times I didn't cry at all. So I was like, cool. So corms on the other hand, so bulb, I put these two together because they're kind of opposites. So if you look at a corm, let me go back to that image. A corm, on the other hand, is almost all stem tissue. When you cut into a corm and look at it, almost all of it is stem tissue. And then there's what you see on the outside, it just kind of looks really papery and thin. Those are the actual leaves. And then, of course, the root. Oh, I guess it is here too. And then here's the bulb with the stem.
That's the difference between garlic and onions.
Yes, exactly. Garlic and onions, that's a good one to think about.
The leaves like the paper part.
Yeah, the really thin papery stuff are the actual leaves. All right, what else do we have? So stolons, okay, here on top, figure 8. So stolons are modified stems that go horizontal over the surface of the ground. So their stolons technically are above ground. Although I have seen some people define it differently, I go with stolons are above ground. And then rhizomes are modified stems that are below ground in the soil. Okay, so stolons above, rhizomes below. And here you're seeing a ginger rhizome. So when you go buy ginger at the store, it's actually modified stem tissue that you're eating or making a tea out of or whatever. Where's the other one? So iris and ginger rhizomes. So it's stem modified, they grow underground and they're enlarged for storage of nutrients and water. Yes.
Bromeliads, where you have to do only cuttings, is that just the rhizomes that we're talking about? Where you can't do any water modification?
Yeah, I think it's the rhizome they're asking you to focus on. I don't think I have a picture of anything similar. No. Tubers, so you'll look at tubers as well. I think you've already looked at tubers in some regard when we looked at starch. So tubers are modified rhizomes, which are modified stems. So you got this idea, if you grow potatoes, where you dig up the root system, and then you'll see these underground rhizomes that have enlarged into tubers, okay? And the white arrows here in the lower picture, that's actually pointing to the lenticels of the tuber. And then this one's sprouting. So, you know, where you see the eyes of the potato, that's actually node tissue. So this is a modified stem. When you eat potatoes, you're eating mostly modified stem tissue, and the skin is actually bark. So you actually eat bark when you eat, if you should eat your skins, by the way. I tend to do that. Even when I make mashed potatoes, I still leave the skins on and do that. Because that's very, very healthy. And if you ever want to grow your own potatoes, you just need some chunk of potato that has some nodes, and that will sprout and grow a brand new potato plant. So you're eating modified rhizomes.
Skin, bark,
Yes,
eyes,
are the node
are
region of the stem.
So those are like the itty bitty bits where it's sprouting off, would those just be like quote unquote branches?
Yes, yeah, it's like looking at a fat branch. Really. Yeah, it's really cool. How about this one? Would you want to eat it?
No.
Sure,
I bet you know what it is.
it's cooked up,
I
like that nice deep fried potato skin.
So this is potato scab disease. And this is a large part of what I was doing with my postdoc. We were trying to solve this issue. And anywhere potatoes are grown, they're grown globally, this pathogen is there. And there's currently no plants, no cultivars of potatoes that are resistant to it. But we found a big group of different Streptomyces bacteria that can kill the pathogen. And we had really good success with field trials. I mean, Frito-Lay was actually they wanted us to keep going. They gave us some of their property and let us do some trials on their land. But this wouldn't kill you. If you did decide to eat it, it wouldn't kill you. In fact, I've eaten it. I don't recommend it.
Things bad, but they
Yeah.
don't just look bad.
So now I'm guessing some of you in here like the smell of wet soil, right?
Yeah.
After a rain, isn't that nice? It's, you know, you go out walking in the rain and that lovely wet soil smell. That's how that tastes. So you bite into it, and it tastes like wet soil. Now, I love the smell of wet soil. I don't like the taste of it. But the reason it tastes like wet soil is that a pathogen that is attacking is one of the bacteria that produces the wet soil smell. And so it's a chemical, one of the chemicals is called geosmin. And that's the volatile that you smell after rain has soaked the soil. And so that's why it tastes like wet dirt. So my postdoc advisor, we had a potato potluck party, a PPP, potato potluck party at her house. And so we all had to bring a dish that was potato based. And then she brought out some scabby potatoes and said, I dare ya. So we all took a bite. And that's when I said, okay, one bite's enough, I'm done. So, but yeah, it was a bonding opportunity for all of us. We were all new to the lab, so. No, she had some raw and then she cooked one.
Okay.
And it still tasted bad, no matter what. I don't know, maybe some spices, if you douse it in spices, maybe, but.
Spice, not thick.
Some other modified stems, so rosette plants. So when you see a plant where the leaves are attached to a very short stem, they call those rosette plants. And so the stems have been modified to be very, very short. Some stem tissue will form into thorns. And then we're going to talk about thorns versus spines, because when we get into leaves, we'll talk about spines. But so you have thorns for protection. So this is, you don't have to write all this down, it's in the PowerPoint, but these are what we've been covering. So really, I mean, often, you know, a lot of people don't give stems much thought, but actually there's a lot of different things that stem tissue can morph into depending on the needs of the plant. And then in your book too, please read the section on the economic importance. Again, most people don't really give stems much thought, but boy, do we get a lot of stuff from stems that we make use of. So of course, wood for all kinds of things, including fuel, but also for building. Charcoal, if you ever picked up a bag of charcoal briquettes and read the side little story, I think they still have the story on the side of the bag of, was it Henry Ford? Henry Ford's son-in-law, I think, came up with charcoal briquettes because back in the day when your dad, probably your grandpa, if he ever had a woody car where the station wagons had wood paneling on the side. That was, the Henry Ford company. I don't know if they came up with that model, but anyways, their company would make that, and then they'd have all this leftover lumber. And the son, I think it was the son-in-law, went to Henry Ford and said, we should do something with this to capitalize on this instead of just throwing it away. And so he developed charcoal briquettes to grill. So hopefully that story is still on. I was waiting in line trying to buy some a few years ago and I was bored. So I started reading that and I'm like, oh, that's so cool. So anyways, ropes and fabrics can get manufactured from the nice secondary xylem, and sugars and spices. Certainly we can get off of some species. Cinnamon, right? Love cinnamon. So that is a famous one. Dyes and drugs and tannins, so all kinds of uses. So your book gets into this. I'll be asking about stuff like this on the exam. So make sure that you give it some thought. I want you to think about all the wonderful things that we've used from this part of the plant. So lots of uses. Even cork, if you're over 21 and have opened a wine bottle with cork stopper, they actually harvest, they have plantations of cork oak trees, and they can sustainably harvest without girdling or causing the plant to die. They'll take off the bark and get to the cork cells of the bark and make the cork. Now, a lot of the cork in wine bottles and other bottles, it's synthetic. It's not cork cells anymore, but there's still quite a few. So here's a cork oak tree and they're harvesting. And believe it or not, that does not kill the tree. So I don't know, I've never looked into the details of how they do it, but they must do it and keep the phloem intact. Otherwise,
It's very gentle.
yeah, they must have a really cool technique so that they're not stripping or girdling the stem. So, and then some of the amber that you might see for purchase, a lot of jewelry is made out of amber. It's just resin coming from mostly stem tissue where it's oozed out. Here you can see a fly got captured and can be very beautiful. Yes.
Is that be a silly question, but is that like sap?
It's a resin that oozes out. What's
It's the sticky stuff from the pipe.
yeah, like the pine trees, where there's like little bubbly areas and you poke at it and this oozy stuff comes out and it's very fragrant. It's kind of like that, the resins that come out. And then they get fossilized over, millions of years. Jurassic Park ideas. And then bristlecone pine, which doesn't look like much of a tree, but in California, I think this is an actual picture of Methuselah, which is the name they've given the tree. And they claim that it's 4,723 years old. Over 4,700 years old.
They have to take a sample.
Yeah, I think they did a core sample. So as far as I know, only a handful of people know where this is because they're so worried that people will go and take samples, as a memento or, trophy or whatever. Yeah, so they keep it hidden, the location. So hopefully that's still the case. But over, 4,700 years old. That's just stunning. And then can't even get it into the frame. That's not me, but that's a huge stem. So now this is a giant redwood. I have not seen the giant redwoods yet, but I plan to. I've seen the giant sequoias. Giant redwoods, anyone got a chance? You out west?
Yeah.
Yeah,
Yep,
Nice.
Cross Bridge, San Francisco.
nice.
Like the Moyer Woods.
Oh, yeah. I was close. I went to Yosemite National Park after a plant biology conference. And we were close, but we just couldn't fit in both. So we had to pick either the redwoods or the Yosemite excursion. And my sister was with me and she's like, I want to see the mountains. I'm like, well, I want to see the trees. So we compromised. She says, well, there's giant trees at Yosemite. And I'm like, oh, okay. So we went and it was awesome. Giant sequoias are awesome, so, but I mean, pictures don't do this justice. If you ever get a chance to see the sequoias or redwoods, highly, highly recommend it. It's really transcendent to walk amongst those types of organisms that are living for so long. So this is one that fell over. The Yosemite, when I went there, they had just redone the area where you can walk amongst the giant sequoia, and I really liked what they did. They moved the walkway much further away from the trees so that because if you think how many millions of people go there on an annual basis, well, maybe not millions on an annual, but a lot of people go there, and if they're all walking right over the soil where the roots are, over time, that's compacting the soil, and that's not healthy. And so they moved the boardwalk and just were more, and then they put a lot of more nice signage up, like science stuff where people could learn, so it was really nice. And then, of course, your giant saguaros. When I was out in Arizona, I went to Las Vegas for a conference. I don't like Las Vegas. I'm sorry if you're from there, but I just had a really good, I guess my experience was the conference was in a hotel conference complex. And you had to, like, you walk into the hotel, the lobby was a casino essentially, to check in. And then they force you to walk through more casino area to get to the conference. And it's all artificial light, no clocks. They don't want you to know what time it is. You'll just keep gambling. And just, everything artificial. And the one thing I did do was go to the botanical garden there, which was lovely. And a chocolate factory. There was a chocolate factory out there somewhere that I went to. So that was really good. So, but here again, here's the giant sequoias. Well, that's one of them I got to see. But it's just the pictures, you can't do it justice. That's another one. Just amazing. And I gotta do this. I don't know if you can see, they're riding horses. It's like, are you kidding me? I could have done that. And I gotta tell my sister, we could have done that. Yes, up in the Keweenaw. Yeah,
It's the closest I've gotten to.
yes. And that's what kind of inspired me. I went there. So that's it's kind of past Calumet, isn't it? Or near
I
Calumet,
don't remember.
somewhere.
A bigger field trip.
Yeah.
Like,
Yeah, has anyone else gone to Estevan Pines? And they have white pines there that are quite, yeah,
we have us adults.
yeah. So if you're jonesing for, something, similar, Estevan Pines is, as far as I know, the closest.
Just something random that I noticed on your list earlier, where it says about like stems and their use. In Mexico, paddle foods are a very common dish to eat, so they
Yes.
even soups and tacos too.
Yeah, the Claddafield, right? Yeah, I saw a recipe with that. I'm like, ooh, yeah. You're okay. Okay, so I'm not missing too much. But yeah, so it's amazing. There's, the medicinal and for food too, stems can be used. So yeah, there's something else. Okay, so yeah, I really do encourage, if you get the chance, to go. Estevan Pines is a good close thing to go see. Those are impressive. And I love white pines. White pine trees are really, really cool. Okay, so stem tissues. Now I want to shift into leaf tissues. But before we do that, let's do a Google form. And let's see if this will work. So you should see two questions with images popping up, asking you to identify two figures. Are you getting that?
Yeah.
So you can chat with each other and look at your notes. This is, let's see what we come up with.
And then it in the stem, it goes up to its guy.
There was. Yesterday. The staff.
Yeah.
All right. Awesome. That's what I gotta do.
Yeah.
Oh, yeah. That works.
All right. Have we all chimed in on our answers yet? Few more people need to chime in.
Yeah.
So almost pass.
Have we all chimed in? Okay,
But it's helpful. So he doesn't. I was wondering.
so which one is a root?
Hey.
Oh, no.
Don't give us that look.
I just love the questioning tone. Call the roots.
Okay.
Okay. Question.
So why? Why would that one be a root? The ring.
You don't need that much support on the outside of your ground.
Yep. So if you're underground being packed in by soil, you don't need to put your strong vascular tissue, such as ground, you don't need to put it out there.
That's what you.
So which one is a dicot? Very good. So dicot what? Yes, dicot stuff. Very good. So this is A root, what kind, monocut or dicot? Good. Mono for a single ring. Root tissue because it's further in. Here you have stem tissue because the vascular tissue is further out in the fibers, so you have the xylem, which are really strong in fibers. So if you're a stem, put all that around the perimeter to give you a little bit more support up in the air like that. Okay. Well, let's start leaves on Friday since we're just a couple minutes. Let's do that.
Quick question. So that was doing the quiz yesterday, and there was some questions that I honestly Googled one of them, and I'm not really trusting it because it's Google, and I don't recall reading anything about this.
Okay,
It says, what is the source for PEP?
Yeah.
They're both C4 and CAM. I don't recall any other information.
Okay.
No, we did it.
so let me show you. It's in the PowerPoint, but you have to,
Oh,
Yep.
I read my notes like based on the PowerPoint and I didn't find anything.
Okay.
but-
let's look. So. Where did it go?
I don't think I remember any white things I bought. See what I want.
Yes.
All right,
I was actually looking at that slide for that.
maybe it's not in this. Where did it? Here, I'm looking at,
I hope gone there with my family, but my sisters are not like outdoorsy people. They're not like the kind of people who are like, let's go to the national parks for the summer. We did not do that once.
Yeah.
So annoyed the whole time, and I was like, guys, that's not.
I went to Yellowstone too, but I didn't.
Outside, and I was like,
so if you look carefully. The
Oh,
source
that
of
was
PEP,
large.
yeah,
Okay, so I have pyruvate for the C4. Would that be correct? It just sounds wrong for some reason.
so you're looking back at the C4,
Okay. Then it's probably that.
the source of the pyruvate. It's actually, if you look here, it's malate coming through when it donates off the CO2,
Mhm.
then you're left with pyruvate. So it's really coming from the breakdown of malate.
Oh, okay, I was looking more like written stuff and not really the pictures, because I write basically like whatever's in the notes.
Yes,
What's actually written, but
Yeah.
I'll check the pictures more into detail.
Yeah.
Awesome, thank you.
Just double checking, it's still good for.
Yep.
Okay, you have